Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I will be going over ketone body metabolism. Now the ketone bodies which are used as fuels for our energy purpose in some of our cells, especially during fasting and starvation. Now as in the figure, whenever fatty acids are coming out of adipose tissue, especially during fasting and starvation, so the free fatty acids which are flooding into the blood carried to peripheral tissues and also liver. So the fatty acids reaching liver will undergo beta-oxidation process to give acetyl-CoA, and these acetyl-CoAs initially in the liver can go into TCA cycle for complete oxidation, but when the TCA cycle is saturated, at that time acetyl-CoAs are diverted into ketone body formation. Now once the liver makes ketone bodies, these ketone bodies are sent out into the blood for peripheral tissues, that is extrahepatic tissues, to use it. So the ketone bodies that are entering into the blood, so since they are water soluble, so we have two ketone bodies which are water soluble. One is acetoacetate, other is β-hydroxybutyrate. So the acetoacetate and β-hydroxybutyrate can also be filtered by the glomerulus and appear in the urine. So in the urine we will find acetoacetate. Let me write it down. So acetoacetate, this is a type of ketone body which you can find in the urine. Other is the β-hydroxybutyrate. These are the two ketone bodies that we will find in the urine sample. And the acetone is a volatile molecule, so it will be exhaled in the breath through the lungs. Now this acetoacetate and β-hydroxybutyrate which are there in the blood, they can be taken up by the extrahepatic tissues, and in the extrahepatic tissues acetoacetate and β-hydroxybutyrate can be oxidized into acetyl-CoA, and the acetyl-CoA can get into TCA cycle of extrahepatic tissues and will be completely burned into carbon dioxide. And this is what in brief about how ketone bodies are synthesized and how they are sent out into the blood, and then how they are used by the extrahepatic tissues. Now let's get into synthesis of ketone bodies. So the synthesis of ketone bodies are referred as ketogenesis, and the ketogenesis will be going on in the hepatocyte mitochondria, and the condition for ketogenesis should be a low insulin and high glucagon levels. That means insulin to glucagon ratio should be low. So during this condition, free fatty acids will flood into the blood and carried to the liver for oxidation. Beta oxidation will give acetyl-CoA, and acetyl-CoA will be diverted into ketone body formation, and that is done by one of the regulated enzyme called 𝗂𝐻𝐵-CoA synthase. One of the important enzyme in ketone body synthesis is 𝗂𝐻𝐵-CoA synthase, which is located in the mitochondria of liver. Now the hepatocyte mitochondrial 𝗂𝐻𝐵-CoA synthase is quite different than the cytoplasmic 𝗂𝐻𝐵-CoA synthase. Now the 𝗂𝐻𝐵-CoA synthase that is present in the cytoplasm is involved in cholesterol metabolism, whereas the mitochondrial 𝗂𝐻𝐵-CoA synthase, it is involved in ketone body synthesis in the liver. Make sure you know that. Now there are three ketone bodies that will be synthesized in ketogenesis process. One is acetoacetate, other is β-hydroxybutyrate, and the third is acetone. And then when you compare between acetoacetate and beta-hydroxybutyrate, so the beta-hydroxybutyrate, which is also referred as 3-hydroxybutyrate, is synthesized much more than the acetoacetate. And also, as I said earlier, acetone is volatile and we exhale it out. Now let's see the synthesis figure here, the flow chart. So Two acetyl-CoAs in the liver, mitochondria of the liver will condense with one another to become acetoacetyl-CoA, and now that acetoacetyl-CoA will be condensed with one more acetyl-CoA molecule with the help of 𝗂𝐿-CoA synthase enzyme to make 3-hydroxy-3-methyl-glutaryl-CoA, and 3-hydroxy-3-methyl-glutaryl-CoA is referred as 𝗂𝐿-CoA. Now the 𝗂𝐿-CoA lyase enzyme is going to break this into acetoacetate, that is one of the ketone body, and this acetoacetate further it will undergo reduction into beta-hydroxybutyrate, because the beta carbon which has got a keto group in acetoacetate is now reduced into hydroxyl group done by beta-hydroxybutyrate dehydrogenase. The reason for conversion of acetoacetate into beta-hydroxybutyrate, although beta-hydroxybutyrate is not a true ketone body, because beta-hydroxybutyrate has got a hydroxyl group there, so it is not really a true ketone body there. But still acetoacetate is converted into beta-hydroxybutyrate. The reason for that is NADH is oxidized into NAD plus, and much needed NAD plus in the mitochondrial matrix is regenerated here, and the reason that is done by beta-hydroxybutyrate dehydrogenase. So because of the necessity of NAD plus, so most of the acetoacetate that are made in the mitochondrial matrix will be converted into beta-hydroxybutyrate. So with that, for every one acetoacetate there will be 4 to 5 beta-hydroxybutyrate molecule, 4 or 5 beta-hydroxybutyrate molecules. So that's what is the ratio. Now, so whenever beta-hydroxybutyrate dehydrogenase enzyme is saturated, at that time some of the acetoacetate will be diverted into acetone by a spontaneous decarboxylation process. So this is how 3 ketone bodies are synthesized in the liver. One is acetoacetate, other is beta-hydroxybutyrate, and the third is acetone. Out of these 3, acetoacetate and beta-hydroxybutyrate are water soluble molecules, whereas acetone is a gaseous molecule, so it will be exhaled. Okay, so for every one acetoacetate you are going to find 4 to 5 beta-hydroxybutyrate in the blood. Now, when the ketone bodies are released into the circulation, so these ketone bodies are taken up by the peripheral tissues. So in the peripheral tissues, what will happen? Like extrahepatic tissues, all extrahepatic tissues other than liver and red blood cell will use ketone bodies. So ketolysis needs one of the important enzyme in these tissues called thiophorease, and this thiophorease is absent in the liver. And RBC cannot use ketone body because ketone body utilization needs mitochondria, and RBC do not have mitochondria. Now the complete oxidation of beta-hydroxybutyrate, effectively it gives 21.5 ATPs, whereas complete oxidation of acetoacetate in the peripheral tissues, that is extrahepatic tissues, will give 19 ATPs. So we can understand that from this figure here. As you see, beta-hydroxybutyrate, it is undergoing oxidation to acetoacetate. During that process, in the extrahepatic tissues, NAD is reduced into NADH plus, and one NADH plus there, it is 2.5 ATP. Now, acetoacetate will be converted into acetoacetyl-CoA. Basically, CoA is added here, and the donor of that CoA, it is coming from succinyl-CoA. Note that the ketolysis, that is breakdown of ketone body, is going on in the matrix of mitochondria. So succinyl-CoA is the one that donates CoA here to convert acetoacetate into acetyl-CoA. So succinyl-CoA in the mitochondrial matrix, it's a TCA cycle intermediate. So that means TCA cycle intermediate succinyl-CoA is diverted here into this reaction. Otherwise, in the TCA cycle, succinyl-CoA could have been converted to succinate by succinate thiokinase enzyme, and there a substrate level phosphorylation could have given 1 GTP. So, and that 1 GTP is not given here because we have bypassed the reaction. That means cell is losing 1 GTP, and which is equivalent to 1 ATP. So, for that, what we will do is we just take out one GTP from here. So minus 1 ATP, I'll just write it as ATP, minus 1 ATP. Okay. Now, once you get activated acetoacetyl-CoA, and it will undergo thiolase reaction where CoA is added and it breaks into 2-acetyl-CoA molecule. And these 2-acetyl-CoA molecule goes into TCA cycle and will give 20 ATPs there, because 1-acetyl-CoA is worth 10 ATP, so 2-acetyl-CoA it is 20 ATP. So now when we calculate a net number of ATPs that you get from oxidation of β-hydroxybutyrate all the way into carbon dioxide, so you're going to get 2.5, that is from this NADH plus, and then you get 20 ATPs from 2-acetyl-CoA, so that is 20 there. So overall you get 22.5, but you need to take this 1 ATP out from it, so minus 1 ATP, that will be 21.5 ATP. So this is what is the net number of ATP that you get from oxidation of beta-hydroxybutyrate into carbon dioxide. Now what about oxidation of acetoacetate? That's another ketone body. So the oxidation of acetoacetate into acetoacetyl-CoA, consuming 1 ATP there, and acetoacetyl-CoA breaking into 2-acetyl-CoA molecules, so you get 20 ATP. So 20 minus 1 ATP, so it will be 19 ATPs. That's what is the energy coming from oxidation of acetoacetate in the extrahepatic tissues. Now the regulation of ketone body synthesis, it will be regulated by insulin-glucagon ratio, especially when there is a decrease in insulin-glucagon ratio. Meaning, whenever there is increase in glucagon, so glucagon, it is going to promote fatty acid oxidation, because glucagon is going to maintain low melanyl-CoA levels, and this low melanyl-CoA level, it is going to free the carnitine palmitoyl transferase 1 enzyme, because you know whenever there is high melanyl-CoA has got a negative effect on CPT1, that is carnitine palmitoyl transferase 1, which is allowing fatty acids to go into the matrix of mitochondria for beta oxidation. So higher glucagon means lower melanyl-CoA levels. Lower melanyl-CoA levels means its negative effect on CPT1 is not there, so CPT1 is free to work, so it is allowing a long chain fatty acids to go into the matrix of mitochondria, and that increases beta oxidation. That's how higher glucagon levels will increase beta oxidation by freeing a CPT1 over outer mitochondrial membrane, and that is done by decreasing melanyl-CoA levels, because melanyl-CoA is a negative modulator of CPT1. That's one thing. Second thing is, whenever this beta oxidation is going on, so there will be higher levels of NADH plus, and that higher NADH plus, what it does in the TCA cycle, it is going to convert oxaloacetate into malate. Okay, so oxaloacetate is all converted towards the malate to regenerate NAD plus, because conversion of oxaloacetate into malate, during this reaction NADH is converted to NAD plus. So because of this, so malate will increase, and this malate will go towards gluconeogenesis to make glucose. That's what happens to malate. So that means there will be decrease in oxaloacetate, right? And this decrease in oxaloacetate means it is not available for acetyl-CoA to combine with, to condense with and make citrate, because in the TCA cycle oxaloacetate is going to combine with acetyl-CoA to make citrate, and now because of decrease in oxaloacetate, so this reaction do not go on. That means oxaloacetate do not condense with acetyl-CoA, simply because there is less oxaloacetate there, because most of the oxaloacetate is diverted into malate to go into gluconeogenesis. So less oxaloacetate means less acetyl-CoA is condensing to make citrate. So at that time this acetyl-CoA is now diverted into ketone bodies, because TCA cycle is not running, so ketone bodies, so there will be increase in ketone bodies. That's how acetyl-CoA is diverted towards ketone body by decreasing oxaloacetate levels. And why oxaloacetate in the mitochondria will decrease? Because most of the oxaloacetates are diverted into malate formation, and this malate, because to regenerate NAD+, and malate goes to gluconeogenesis, while lack of oxaloacetate will lead to elevation of acetyl-CoA, and that acetyl-CoA is now diverted into ketone body formation. So like this, glucagon will make sure, so malonyl-CoA levels are low, CPT1 is active, beta-oxidation is going on sufficiently, increases the NADH level, and that increase in NADH level will divert oxaloacetate, which is required for TCA cycle to continue. Oxaloacetate is diverted into malate, thereby it is not available for condensation with acetyl-CoA to make citrate in the TCA cycle. So the acetyl-CoA will build up in the lack of oxaloacetate. So this acetyl-CoA now, because it's not going into TCA cycle, it is just going into ketone body. So it is diverted. Acetyl-CoAs are diverted into ketone body formation. This is a kind of regulation that will be going on during fasting condition in the hepatocyte mitochondria. Remember that. So that's what is shown here in the figure. So whenever fatty acids are raised, so these fatty acids, and especially when glucagon levels are high, so malonyl-CoA levels are kept low, and low malonyl-CoA means CPT1 is active, and when the CPT1 is active, so the beta oxidation is going on, and this beta oxidation is going to increase acetyl-CoA levels, and acetyl-CoAs, increased acetyl-CoAs, so their entry into TCA cycle is limited because it needs oxaloacetate. Acetyl-CoA needs oxaloacetate to combine or to condense to make citrate. Okay, so now what happens to oxaloacetate? Oxaloacetate is diverted into malate side, malate formation, because to regenerate NADH plus H plus into NAD plus. So oxidation of NADH H plus into NAD plus is going on to regenerate NAD plus. So oxaloacetate is not available sufficiently to condense with acetyl-CoA in the TCA cycle. So in that case, what happens? So acetyl-CoA builds up, that's what is shown here, and this acetyl-CoA now it is diverted into ketone body formation. So like this, under fasting condition, specially mediated by glucagon, so ketone bodies are increased. So it is all starting with low levels of malonyl-CoA maintained by glucagon, and low levels of malonyl-CoA means higher activity of CPT1 and allowing fatty acids to run down into the mitochondrial matrix for beta oxidation and increasing the acetyl-CoA levels. The reason for increase in acetyl-CoA levels means because TCA cycle is not going on efficiently, because lack of oxaloacetate here, and because why lack of oxaloacetate? Oxaloacetate is all diverted into malate, which is going towards gluconeogenesis. So in the absence or lack of oxaloacetate, acetyl-CoA simply builds up instead of going into TCA cycle to make citrate. So acetyl-CoA is diverted into acetoacetyl-CoA and later into ketone bodies, that is acetoacetate, β-hydroxybutyrate and acetone. So this is how a regulation of ketone bodies will go on. So that's about regulation of ketone bodies, ketone body synthesis, ketone body degradation. So that's about it. Thank you.